Hello, everybody. My name is Dan Geschwind, and I'm the director of the Institute for Precision Health and a professor in neurology, psychiatry, and human genetics. And it's really my great pleasure to introduce my colleague and my friend. I hope he considers me that. I consider him a friend. To introduce Bogdan Pasaniuc for the CTSI Distinguished Speaker Series. Bogdan is an associate professor of, aside from having one of the more difficult names to pronounce, he's an associate professor, for me, he's an associate professor of computational medicine, human genetics, pathology, and laboratory medicine, where he was originally hired into. And he's the associate director of the Bioinformatics Interdepartmental Ph.D. program. Although he is one of our own, and I think there is sometimes a tendency to take our colleagues for granted at some level more than we should, he is one of the people who at least those close colleagues of his do not take for granted. He is well recognized within our community and outside as a visionary thinker, a leader in his field of statistical genetics and computational medicine. He completed his Bachelor of Science in Computer Science in Romania in 2003, a PhD in Computer Science and Bioinformatics at University of Connecticut, and subsequently a 3-year postdoctoral fellowship at Harvard School of Public Health and MIT Broad Institute. He was hired to come to UCLA in 2012, where as an assistant professor in pathology and in human genetics, where he's had major influence on many programs. I'm just going to say a couple things. Here at UCLA, we're gifted with an exceptional bioinformatics graduate program. And Bogdan is one of the people who's really, along with his colleagues, and there are a lot of them, have played a major role in building it to be a world-leading program. And what makes Bogdan truly special is what all of us recognize as his collaboration. He's super unassuming, easygoing, but this is linked underneath with an exceptional creative drive. And it's in this capacity that he pivoted his work to begin to play a major role in the Institute for Precision Health. I would say relatively selflessly creating resources and building platforms with some of the other faculty who were involved, as well as doing a lot of QC on genetic and clinical data, and as well as playing a critical role in linking our work here at UCLA with many international consortia. And one example of that is his work with Manish Butte in COVID host genomics infrastructure, in which he's playing a major role in connecting us with the Biobank and Common Genetic Host Genomics consortia. His work is having major influence in human genetics broadly. His 2016 paper on transcriptome-wide association that was published in Nature Genetics has been cited over 600 times just in the last few years. And he's beginning to apply these same types of innovative approaches to UCLA medical records, leading to the genesis of many important studies that I think he's going to talk about today. So it's really with great pleasure that I introduce Bogdan Pasaniuc.
Thank you so much, Dan. I hope I live up to your wonderful introduction. Let me see if I can share. Okay. Okay, so hopefully you guys see my slides today. So I'll be talking today about a bunch of methodologies or projects where we're trying to leverage, as Dan mentioned, some of the wonderful resources that we're building here at UCLA that combine electronic health records with genomics within the UCLA Atlas Biobank. And for the first half of the talk, I would spend a little bit of time describing the resources that are being built there. And you can think about it as an invitation for all of you that are on the call that are interested in working or collaborating with us to extract more information from all these resources. And in the second part of the talk, I'm going to describe some of the essentially vignette type of projects that I hope can convince all of us that genomics can play a role as we try to improve patient care in precision health. And I'll start by just acknowledging, some of the grad students that I'll be presenting their work today. As Dan mentioned, we're blessed here with a wonderful PhD programs, and some of the students or the trainees that have played a key role in all the projects that I'm going to present today are Malika Frond, who's a PhD student in genetics and genomics, was a PhD student in genetics and genomics. She just graduated this summer, and she's now a genetic counselor at Stanford. Ruthie Johnson from computer science, a PhD student program, and then Tom Areni from the bioinformatics PhD program. So to get started, as we think about the role of genetics or genomics in precision health or in general in human diseases, there are two ways to think about, or essentially two ways to think about human diseases. On one side, and this has been the traditional way where we can think about the genetic contribution to human diseases. On one side, we can think about rare Mendelian phenotypes that are where genetics plays a major role in driving the effect size for those diseases, where mutations in key genes have very large effect, large penetrance. They typically affect one gene or a few genes, and because they have a lot of deleteriousness, they are kept at rare frequencies in the population, and they usually lead to Mendelian forms of different diseases. On the other hand, there are the standard common diseases or complex phenotypes that range in terms of how much of the genetic factors contribute to their risk. And the genetics of these variants typically have small effects. They affect many genes on the order of thousands, if not tens of thousands, for the super highly polygenic phenotypes or diseases that we study, and that they are usually common in the population. And this traditional view of human diseases has led to breakthroughs in terms of genomics in the past 2 decades. On the Mendelian phenotypes has been one of the most exciting breakthroughs, where genomics has quickly gone from a research setting where genes were discovered for different Mendelian forms of diseases, all the way to being currently the de facto method to clinically diagnose patients with rare disorders. And I'm listing here some relatively recent papers, and I'm highlighting a relatively new center that just was opened up in the past couple of years here at UCLA, the California Center for Rare Diseases, that's leveraging a lot of the foundational work that Stan Nelson and his colleagues here at UCLA through the Undiagnosed Disease Network have been essentially pushing genomics from a research tool from Mendelian phenotypes towards actually being used on a day-to-day basis in clinical setting. In contrast to Mendelian phenotypes, the complex phenotypes like heart disease, let's say risk for cancers, all sorts of things that affect a large proportion of our patients, the genomics for them has not been as successful as for Mendelian phenotypes. Within the past 10, 15 years, the field has found tens of thousands of regions in the genome that contribute risk for these diseases. And I'm highlighting here a brief review a few years ago that showcases the rapid pace of discovery. On the x-axis, you see years, and on the y-axis is counts of regions in the genome that contribute to this common diseases. And you see that it's on the order of tens of thousands of regions in the genome that contribute to traits like prostate cancer, breast cancer, schizophrenia, and so on and so forth. To first approximation, virtually any common disease in humans has been studied by one of these large-scale genetic genome-wide association scans. And now we have a catalog of all these findings. We're not there yet in terms of how we translate this to the clinic. And this is probably one of the big open areas of research in the next 5-10 years in terms of genomics for complex traits. So as we think about how these genetic discoveries get propagated towards clinical setting, towards improving patient care, we require, in medical settings, we require the genetics of our patients for translating these genomics findings. And within the past 5-10 years or so, most medical enterprises have started creating biobanks where the genetics of patients in the system gets stored and gets generated on the level of either genotyping arrays, which I'm going to go a little bit into, or a whole genome or exome sequencing, and they are linked with their entire medical record or the electronic health record, EHR. Here I'm showing you a brief review of most of the biobanks that currently exist worldwide. And on the x-axis, you see the year enrollment. On the y-axis, you see the proportion of non-European individuals in that biobank. So on the bottom axis, you see a lot of European biobanks that got started, visionary got started maybe even 20 or so years ago, like the Decode in Iceland, or the Estonian Biobank, UK Biobank, and so on and so forth. And on the top, you see biobanks that got created relatively fast, and they expanded really large within Asia. And somewhere in the middle, you see a lot of the biobanks that are currently being built in the US across multiple medical systems. And somewhere there, somewhere in 2016-17, UCLA Precision Health Institute created the biobank, and I'm going to describe a little bit more about this today. So I'll start my talk today by just giving you a brief overview of some of the resources that a lot of folks have put a lot of effort in the past 3, 4, 5 years at UCLA to create ways where we can first improve research outcomes, and second, quantify how will we translate these genomics into clinical care. So the two resources that I'm going to focus today is the data discovery resource, so-called DDR, and the Atlas, which is the genetic variant of the DDR. So just to highlight what this data, what the resources are, you can think about them as the DDR represents a copy of all the electronic health records at UCLA that has been de-identified, and it's been used for research. So there's all the personal identifiers have been stripped from the data. It contains a lot of demographics for all of our patient population, billing codes, lab tests, medications, all sorts of visit information, and so on and so forth. And because the electronic health record was introduced at UCLA around 2013, we have data within the DDR starting from that time onwards. The number of records or number of patients that get touched by this DDR are around 5 million, but after some initial quality controls or restricted to patients that are being in the system for a few years or have enough data to look for whatever research questions, we're looking at the order of 1 to 2 million records that are we can do research on them. And attached to the DDR, a flagship initiative of the Institute of Precision Health here at UCLA has been into creating a biobank where a large proportion of our patients get consented, and their DNA gets stored in a biobank, and then genotyping data gets generated, and probably in the future some sequencing data as well, to the order of 100. At this time, the pilot project is to generate 150,000 genotypes from 150,000 patients. The current status of that, so the project is ongoing. The current status of that is that around 70,000 patients at UCLA have been consented. As you can imagine, this is a very, very large effort from a lot of folks, from a lot of departments, from a lot of places to consent 70,000 patients within our system. And around 30,000 of them, we already have, genetic data that we can connect with the electronic health records, and we can perform interesting analysis that I'm going to some of them I'm going to show to you today. So this first wave of the data from the October is around 30,000. So how do we look at the data at UCLA? As you can probably imagine, our data is very diverse. I'm showing you here self-identified race ethnicity. And what you can see from this plot, these numbers, that around 30 to 40% of our patient population self-report as being not white or Caucasian or not Hispanic. So there we tap into a lot of diversity, which is, as you might expect, given that we serve Los Angeles, which is a very diverse place. So just extracting very brief demographics from the DDR, we can tell that our patient population is very diverse. So the next question that we ask is whether the genotyping, the patients that we consent for genetic research, whether they mirror the same diversity in terms of self-reported race ethnicity. And to a large approximation, that's definitely the case. So on the left side, you see the diversity from the DDR, so from the electronic health records of around 1 million patients. And on the right side, you see the breakdown in terms of self-reported race ethnicity from the patients that we've genotyped at that time, which is a year ago in the zero wave of the atlas. And the breakdown is very similar, with the exception of the individuals that are unknown. So these are patients that don't self-report anything, and they presumably don't want to participate in genetic research. So with the exception of that, you see that in general, we recover the same diversity with the genotypes as we observe from the electronic health record. So this is based on self-reported race ethnicity. Now we can also infer what is being referred to as genetic ancestry, which it's a concept that is completely distinct from self-reported race ethnicity. So the genetic ancestry reflects something about whether the genome, the genotypes, are similar to other genotypes that have been, generated elsewhere. So what I'm showing you here is a principal components plot that takes genetic data from all of our patients and essentially projects it on some axis of variation, principal component 1 and 2 on the two axes. And we plotted them together with publicly available genotypes from individuals that are from different parts of the world. And every dot there is one genome, so it's one patient. In gray, you see the atlas patients, so around 30,000 points, gray points there. And in different colors, you see it, for example, in the green, those are individuals that were collected in China and Japan, so they're East Asian. And on the bottom, you see European individuals from publicly available sources from the 1,000 Genomes Diversity Project, the African and the Latino individuals in orange and red. So the key take home here is that genetically speaking, the patient population at UCLA that we genotype is very diverse. We see gray dots there across all these major axes of genetic diversity. So we have patients that genetically are similar to Europeans, are similar to East Asians, are similar to Africans. And we can look at, here I'm just showing you the first two axes of variation, the first two principal components, but we can look at all sorts of axes of variation. So we can look at the first two, the first three, and so on and so forth. And each of them tells us a little bit about how genetic ancestry varies within our data and in respect to the data that we have in public resources. We can use this to classify individuals based on their genetic ancestry. And we do that mainly for genetic association analyses. And you can see that the genetic ancestry groupings, which again have almost nothing to do with, or they're distinct from the self-reported race ethnicities. The genetic ancestries, you see that they have a very wide diversity across all these different continental groups. So now we have a way where we can collect large amounts of genetic data. So this is based on more than 500,000 sites in the genome that were genotyped for every patient across 30,000 of our patients. The other component of almost all the research that we would want to do is extracting meaningful information from the electronic health records. And for those of you that work in or have worked or are interested in working in electronic health records and extracting information out of it, you can imagine how complex that is. Electronic health records are an amalgam of lots of data, and they're often in a very unorganized format. So generally, most research gets done from electronic health records by looking at billing codes. So these are ICD codes, and they're typically used in a lot of research as approximations for phenotypes. And the positive side of it is that most institutions are using ICD codes, and most institutions are using the latest version of ICD codes, and they are very detailed in terms of what types of phenotypes get stored in the ICD codes. But obviously, ICD codes are not designed for phenotyping, but they are designed for billing. And you can imagine all sorts of biases across different medical enterprises in terms of what billing codes get used for describing different patient outcomes. So they are very detailed, and for most of the research analysis that we do, we don't really need that level of detailing. For example, there are billing codes for whether the broken foot is on the right or on the left side. There's a billing code, there's an ICD code for getting bitten by a seal, and so on and so forth. And most of the times, they don't really denote meaningful diagnoses that we would want to do research on. So, sometimes they denote activities like cooking and baking, and again, they are being used as proxy for phenotypic analyses, but in general, you observe a lot of biases for doing that. So what we've decided to do is to convert this ICD codes into a more biologically meaningful hierarchy of phenotypes, which are called Phi codes. And these were introduced by our colleagues at Vanderbilt about 6-7 years ago, and in this paper in 2013. And what the Phi codes do, they connect lots of ICD codes into meaningful biologically oriented phenotypes. And I'm giving you an example here where the Phi code that denotes acute upper respiratory infection pulls ICD codes from different parts of the tree of ICDs, all from, and they get grouped together into a more meaningful phenotype that we can analyze and perform research on. And this grouping takes from 68,000 ICDs towards 1,800 Phi codes that are all organized into classes of biologically oriented traits. So let me give you an example of how the Phi codes look like at UCLA. So here's the breakdown in the DDR of 18 classes of Phi codes, from circulatory system to congenital anomalies, all the way to pregnancy complications. And on the x-axis here, you see the number of patients that have those Phi codes. And I know that we have data across 5-6 years of EHR. So a given patient might have multiple Phi codes. So that's why the numbers don't add up to 1.5 million, but they reflect the patient record for any given patient at UCLA. When we look at diagnosis events, we see what you might expect, that certain types of Phi codes have way more diagnosis events than others. For example, if you focus on the respiratory class of Phi codes, there are 500,000 of patients at UCLA that have Phi codes for respiratory traits, but in terms of diagnosis, they're probably more than 2 million, which implies that within the past 5-6 years, every patient that has a respiratory phi code had that phi code on the average 4 times. Obviously, the distribution will be skewed and so on and so forth, but this can be used as a temporal analysis of the data. So focusing more on the classes of phi codes, I'm showing you an example here by diving into one of these classes of phi codes, into neurological traits, and you see what you might expect, that phi codes that reflect chronic pain and so are more abundant than phi codes that are more rare in the patient population. So this tells us a very quick look at how our data within the DDR gets organized. We can just at this stage with the phi codes, we can already start asking questions that might be interesting to dissect. And I'm showing you the distribution of these phi codes by sex and age. Here on the bottom side, you see the distribution by of patients across different age groups on the left side, and by sex on the right side. And you can already see that different types of symptoms or different types of phi codes are enriched in different groups of patients. You can see congenital anomalies are enriched in patients that are on the young side, and this is because within the medical record, if a particular patient gets born, there's a label there about whether there's a congenital anomaly. Similarly, looking for trait that you'd expect to be more abundant in the elderly, like circulatory system phi codes, you see an enrichment in patients over 65. We can also break it down by self-reported race ethnicity, and you can already start seeing things that might be interesting to explore. For example, we see pregnancy complications being more abundant in individuals that don't self-report as white or Caucasian. So we see more pregnancy complications in individuals that self-report as Hispanic or Latino, for example. And again, these are things that we can start diving into it, and those are things that we can easily do on top of this phi code system. We can also do it more statistically oriented, and we can validate that these phi codes truly represent meaningful phenotypes in our data. For example, if we focus on some example traits, we do see that the vast majority of patients that have a phi code of Alzheimer's disease are actually over 65 years old. So this clarifies that the phi code system is capturing meaningful information in the data. We see, for example, that individuals that have sickle cell anemia based on the phi code system are way more likely to self-report as African American. We see an enrichment of autism in males versus females, and so on and so forth. So all these are enrichments that are expected, and they've been published, and we use them to validate that the system of phi codes is actually meaningful in the real data in the DDR. And sometimes we also see things that are potentially novel, like this enrichment of spina bifida, that for which some level of publication exists, but in general, just looking at this correlations between different traits or by demographics, it's a very fast way to ask interesting hypotheses within the DDR data. So hopefully by now I've convinced you that that the DDR and the Atlas are very useful resources, and if you're interested in performing research on them, I invite you to make use in your research questions. So in the second part of the talk, I'll describe some vignette research questions that we're performing on the Atlas and DDR, with the intended goal of asking in research setting whether this data would be meaningful to improve patient outcomes. And I'll describe this in more details for each of these vignette research questions. So the first obvious thing that one could do, now that they're genetics and the EHR, is to ask whether we can recover genetic associations that are well known, or we can identify new ones. So one thing that the linking genetics with biobanks can do, and this is probably one of the most powerful approach in this case, is to take a particular genetic variant and link it to the entire pH code catalog. So you can think about it as a simple way to assign putative function to a particular genetic variant. And this has been labeled as a PHEWAS. So it's a phenotype, association scan. And I'm showing you here with an example for a well-known gene that is a risk gene for skin cancer in our data. So what you see on the x-axis there is different classes of pH codes or different classes of the diseases. On the y-axis is the log 10 p-value. And whatever is above that dotted red line means that it's significantly associated. So we took a well-known gene for skin cancer and we associated across all the phenome in Atlas, in Atlas and the DDR. And as you might expect, a lot of the traits that are neoplasm and they are skin cancer related traits, for example, pH codes like non-epithelial cancer of the skin, squamous cell carcinoma, melanomas of the skin, all show up as significant. So this is reassuring and confirming that this IRF4 is actually a skin cancer gene. And you can also see interesting, potentially new, in this case, not so novel, but you see, for example, actinic keratosis as also being significantly associated with genetic variation next to this gene. In this case, it's not totally novel that the keratosis is, and it actually makes a lot of sense to be significant. But in general, associating a single genetic variant to all the pH codes gives a very nice way to connect genetic data with lots and lots of symptoms in the biobanks. The other way to do it is a more traditional way, where you can focus on a particular trait and then connect all the genetic variants in the genome, in this case, all the common ones, all the 500,000 to 1 million of them that we genotyped on this array, to one particular pH code or one particular disease. And I'm showing you an example of one of this GWAS, or genome-wide association scan for gout in the Atlas data. And you can already see two major hits, which are the hits or the genetic variants on chromosome 4 that are well known to be risk variants for gout. The downside of this is that our data is not as large as the GWAS data that already exists there, but together with all the other biobanks, we've put together an effort to combine data across 20 or so biobanks across the world that share the summary type of data across multiple GWASs to discover potentially new risk variants for all these diseases. So across 20 or so worldwide biobanks, there are about 2 million or so patients for which their genetic data is linked with their EHR. And as a brief kind of a summary, all this work is ongoing together with our colleagues from all these biobanks. And I invite you to hear a talk from Wei-Ju at the next week at the American Society of Human Genetics, where an analysis of about 15 common traits was performed across all these biobanks, and many novel risk variants have been found. And you can see here kind of a breakdown of potentially novel versus replicating no discoveries, anywhere from asthma to appendectomy and so on and so forth, stroke and so on. And this infrastructure across all the biobanks has also been used, and I'm not going to talk about it today, but has also been used as an infrastructure to analyze COVID data. So all this biobanks across the world have patients that have been tested for COVID, and a lot of genetic associations for COVID have been performed. And I invite you a week from now to listen to a plenary session at the American Society of Human Genetics that describes a bunch of the findings looking at the host genetic determinants of COVID-19 risk and severity across more than a couple of thousand, over like 6,000 to 10,000 cases, individuals that have COVID-19 versus a couple of millions of controls. It's also up there in terms of associating genetics with outcomes in this data. And I'll focus next on potential or areas where we think genetics can make their way to the clinic in a fast way. And I'll describe a little bit the case of benign ethnic neutropenia. And this is a fascinating trait in that it's defined as individuals that are asymptomatic, but have lower white blood cell counts or lower neutrophils than let's say the average in the population. So it's benign, so it's asymptomatic. These individuals don't have any other symptoms. So their levels of white blood cell counts or neutrophils are lower than the average in the population. And there's been a lot of research done in this work, and I'm showing you here on the right side a distribution of the neutrophil counts, and it's stratified by the genetic ancestry at a particular region in the genome. So what you see there is that individuals, and in that case, in this data, it's African Americans. So what you see there is that African Americans that have 1 or 2 copy of the European allele at that particular region in the genome on chromosome 1, I think, they have a different distribution than individuals that have 2 copies of the African allele. So the individuals that have 2 copies of the African allele have, on the average, around 1 standard deviation lower neutrophil counts than other individuals. And this has been mapped, and there's been a lot of research done on this. This has been mapped to a particular single nucleotide polymorphism in the dark gene that encodes the Duffy blood group antigen. And the G allele at this particular site is appearing mostly within African ancestry. So this G allele is very low frequency in non-African individuals. And as it turns out, individuals that carry 2 copies of the GG genotype, they have very large effects on developing benign ethnic neutropenia. It's a lower white blood cell count, but with an absence of any other symptoms. So the benign. So long story short, genetic ancestry and this particular SNP, this particular variant, this particular genotype, can be used as a proxy or as a predictor for benign ethnic neutropenia, or it can be used as a marker for identifying individuals that have, lower white blood cell counts than the average in the population, but otherwise, they're normal, or they don't have any other symptoms. So we looked into our data and tried to quantify whether that's really happening in the Atlas data. And first, we looked to see whether individuals that are genotyped, and so this is a previous version of the data, so it's around 10,000 individuals, around 500 individuals have the GG genotype in Atlas, and the vast majority of them are self-reported African Americans, as you might expect, because this variant is high frequency on the African background. And super interestingly, when we looked at the white blood cell counts in the DDR for these particular patients, we do see a 1 unit decrease in the standard deviation, 1 standard deviation decrease in the white blood cell count for these particular individuals. So this variant could be used as a predictor or as a biomarker. Individuals that carry the GG genotype will have low white blood cell counts, and this can be used hopefully in the clinical setting in the future. A different vignette of looking at how having access to a large biobank with diverse set of ancestries would be helpful for improving patient outcomes is into genetic diagnosis using rare variants. So there are a lot of traits or a lot of diseases where genetic variants that are labeled as pathogenic are being used for clinical decision making. And some of these variants are not necessarily pathogenic, or they don't cause the disease across all population groups or across all ancestries. And I'm going to highlight a paper that made its round a couple of years ago, a fascinating paper that looked at cardiomyopathies, where 5 genes are being used, or mutations in 5 genes are being used routinely for clinical diagnosis for this cardiomyopathy, the genetic form of it. And what these guys did, they looked into genetic diagnosis of cardiomyopathy across ancestry groups, and they compared white Americans versus Black Americans in their definition. And what they've observed is that these mutations that are being used routinely for diagnosis have elevated frequencies in Black Americans, sorry. They have such elevated frequencies. For example, the mutations in TNNT2 are on the order of 27% in the population of Black Americans, that are inconsistent with the fact that this is a rare genetic disorder. So the incidence of this cardiomyopathies are rare, or this disease is super rare in both populations. So the fact that these mutations, essentially 1 in 3 individuals that self-report as Black Americans carry that mutation, is inconsistent with the fact that this is a rare disorder. So the most plausible explanation is that these mutations are actually mislabeled as pathogenic, and they're misused to diagnose patients of different non-European backgrounds, and they actually increase misdiagnosis rate in those populations. And what they conclude is that you could, just by sequencing maybe the genome of a couple of hundreds of African Americans, would have detected the fact that these mutations are too high frequencies in those populations to be pathogenic. And this is not just one outlier case. This happens actually quite regularly. For example, in the Exome Aggregation Consortium, they identified a couple of hundreds of these pathogenic mutations that should be reclassified as benign in different population groups. So, taken from this idea, we try to replicate or try to investigate whether we could leverage the fact that the UCLA patient population is very genetically diverse to identify or to maybe localize mutations that could be mislabeled as pathogenic when in fact they are not. So when the Atlas genotyping chip was created, a couple of, maybe around 10,000 of variants that are labeled as pathogenic in the literature were added to the array. And we focus on those for this particular analysis. So we took around 10,000 so-called pathogenic variants that were labeled in the literature as pathogenic or likely pathogenic. For this analysis, we were very, very conservative. So we looked at just the individual mutations, no structural variants. We looked at mutations that are present on this genotyping platform, and we were very rigorous, or we threw away any variant that had any slight potential issues about genotyping error in the data. So the very highly quality calls from the genotype. Then we crossed this mutations, or the frequency of these mutations, across 10 self-reported race ethnicity groups in the UCLA DDR. And we focused on, because we wanted to estimate frequencies, we focused on groups of individuals that are at least 100 individuals in the data that we looked at. And I'm going to highlight just 2 of these mutations. So there are many others that we're diving through it and trying to see what's happening with them. But here I'm highlighting just 2 mutations that haven't been reported so far, where we see their frequencies in the atlas in a particular self-reported race ethnicity. For example, the first SNP, we see it to be too high of a frequency to be consistent with a rare phenotype. And same thing with the other mutation. So we think that by cross-checking frequencies of these supposedly pathogenic mutations with different population groups, we can reclassify different mutations as potentially pathogenic. And the data is gonna seeping through, and that's one of the potential utilities of this type of data. All right, so in the next vignette of this type of research, we also looked at potential utility of incidental findings. So potential utility of, remember, we're genotyping all these patients, and sometimes we might find mutations in genes that might have clinical action. So here, in the research setting, we asked whether or how many of these mutations would we be able to find in genes that are clinically useful. So this is coming from ACMG. There's a list of clinically useful genes, and we focused on 10 of them that are, the top 10 genes with clinical utility. And these are, you probably recognize them: the BRCA1 or 2. There's the ApoB, the hypercholesterolemia, Lynch syndrome, and so on and so forth. So these mutations in these genes are to be reported to patients as incidental findings if they're found. Remember that our data is not, we're not really reporting anything. We're not allowed to, and the data is not really supporting that. But we're asking in research setting, if we genotype 10,000 patients, how many of those 10,000 patients would have mutations in these genes, and whether the patients that have mutations in these genes would actually end up developing the syndromes or the cancers in this case that reflect those mutations. And that's exactly what we did. So we started with 12,000 patients. This is wave 0 of the Atlas data, and we very conservatively reduced the data from 700,000 variants that were genotyped on the platform to about 19 variants in these 10 genes that we can confidently tell. And again, we're very, very stringent here, so presumably there are way more than 19, but again, we just wanted to be very conservative. So there are 19 variants in these genes that have carriers, or at least one patient that has mutations in these genes that are consistent with a potential pathogenic mutation. So what's the breakdown? Well, these 29 carriers out of 10,000, most of them, contain mutation in BRCA1 or 2, and the other ones, and you see the breakdown right here. And the next question that we ask is whether these 29 carriers actually have phi codes that are consistent with this mutations. And as you might expect, very, very exciting, 23 out of 29 do end up developing, cancers or hypercholesterolemia as these mutations would predict. So this is validating that the entire process of, genotyping and looking at all these platforms and the phenotype extraction from DHR actually points us to, accurate data, and these individuals actually carry these mutations. We also see 6 patients that are carriers without this phi code, and these might be, again, in the future, about the yield, this might be patients that would be worthwhile investigating. So I have here an example for this, and I'll just skip through it in the interest of time, but I'm happy to talk more about it if questions arise. So in the last component of the talk today, I'll be talking about a reverse way of thinking about the data. So far I talked a lot about using genetics as the base and then moving towards diseases. So using genetics as as kind of like the entryway towards asking interesting questions in this data. In this last talk, I'm going to talk about doing something reversely, where we look at the phenotype or the diseases themselves, and then look at the genetics as a way to confirm our findings. So this is a collaboration with Manish Bhutt from Pediatrics here at UCLA, and it started by looking at a particular autoimmune disorder called common variable immunodeficiency disorder, which is a very heterogeneous group of disorders that are very rare. They affect around 1 in 25,000 or 1 in 50,000 people. And because it's an autoimmune disorder, patients that are suspected to have this disorder, they have a wide range of symptoms that make them enter the system through a wide array, essential to almost all types of clinics at UCLA. So our goal here was to come up with a computational method that quickly identifies such patients and refers them as soon as possible to an autoimmune specialist to get diagnosed. And as you might imagine, early diagnosis, much more beneficial for patient outcome in the long run. So kind of cutting that time from that could range from a couple of months to a couple of years, while the patient gets kind of bounces around from different clinical specialty before gets diagnosed. Cutting that down to a very short time makes a huge impact for patient outcomes, as well as for if you think about cost reduction, the patient spending years in the hospital it's not cost effective at all. So the goal was to develop an algorithm that leverages electronic health records to figure out quickly patients that are suspected of carrying or being CVID patients, refer them to genetic diagnosis, essentially. So how do we end up doing that? The way to think about this is that every patient might have a lot of features from the electronic health records that are indicative of CVID in this case, or of this rare disorder, but none of these features is actually predictive accurately of this. So none of these features is super precise. But by combining them into a machine learning algorithm, we can figure out, or we can prioritize, we can come up with a risk score that would prioritize individuals that are more likely to carry this disease. So for example, if we restrict to CVID, typically patients with CVID have cough, activity, they have a lot of ICD codes, including IgG levels that are over some threshold. But in general, it's very hard to look at them at one of these features and combine and make a prediction. So what we ended up doing is taking the description of common immune of CVID, convert that into phi codes. So convert that into human ontology terms, which will convert it to a set of phi codes, and you see here a bunch of phi codes there. And after we have these phi codes, ask ourselves what's the best combination of the phi codes that will predict whether a particular individual will have CVID. So all these phi codes, if you look at them, they are quite common in the population, but we want to predict something that is very rare in the population. So that opens up an interesting challenge here. So how do we end up doing it? So you can imagine that we're searching for something that it's very, very rare. So, a fractional percent of patients at UCLA are expected to have CVID. So we're looking for something that it's very few patients in the entire, million patient records might have CVID. And if we look at all these phi codes that are tail marks of CVID, a lot of patients have it. For example, acute upper respiratory infections, 13% of all the patient population have it. They're enriched in CVID, 24%, but as you can tell, just based on this phi code, acute upper respiratory infection, it's not going to be a good predictor for people that have CVID or patients that have CVID. And same thing with all these other potential phi codes that are markers for CVID. However, if we combine them into a machine learning algorithm that takes these as features and puts them into a regression framework, the combination of four could, in practice, identify patients that have either all four of them or a combination of them that are way more likely to have CVID. So without going into lots of the details, we can take data that for all of our patients, all these features that we select for CVID, we can take the CVID status, and we can do cross-validation and train a machine learning algorithm that combines these features together. And the key aspect of this is that we have access to about 200 patients for which we know the ground truth. So we can use them as a validation for this classifier. And we tested a bunch of methods. I'm not going to dive to a lot of the details. One of them that we focused on was ElasticNet, which is just a classifier that penalizes different and assigns different weights to different phi codes. And when comparing to other methods that have been proposed for rare disease stratification, the method PNET is vastly outperforms the proposed methods. And I'm gonna guide you through the fact that the x-axis here is in log scale, and the y-axis gives you the proportion of known cases that are captured by this. So we apply this method, we rank order all the patients, and then we ask, out of the top 100 patients, out of 1 million patients that we apply this, how many true cases are there? And the answer is that 20% of all the cases are for the PhENAT method, while the other method gives you, close to 0. And the interesting bit is that with 1,000 patients, so if you look at 10 to the 3, so the top 1,000 patients already capture 60% of all the known cases of CVID in this data. So that's a massive reduction from, starting from 1 million patient records, reducing it to 1,000. That reduces the workload drastically when we prioritize these patients for potential diagnosis. The other component of the PhENAT that I'm going to just briefly describe this is that there's a temporal aspect to it. Because PhENAT is an aggregation of ICD code, you can compute it on the fly at different time points as new ICD codes get added to a particular patient record. So here's one example for a particular patient, and on the bottom you see the PhENAT score, so the higher the higher risk. And then on the top you see the Phe codes as they get introduced into the medical record for that particular patient. And on the red there, you see, when the ICD for immunodeficiency was added there. So you see that the score, the PhENAT score, was super high and indicative of this patient of having ICD codes, of having a CVID, even 6 months to a year before this patient received the ICD, received the diagnosis for CVID, and was confirmed by the IgG levels. So there's a temporal aspect of applying this computational risk scores, in that they might be able to shave maybe like a year, 6 months to a year from a particular patient's risk to, or potential diagnosis odyssey to get diagnosed for CVID. And as I was mentioning before, we went about for these patients that are at the top risk based on the PhENAT score, and we went back and see whether they have mutations in genes that are involved in CVID or in immunity or in other types of genes. And I'm showing you some of these mutations that we're investigating, but there's nothing really more to say about this. All right, so I've gotten through a bunch of the different projects, a bunch of different things. Hopefully I can convince you that we have a lot of interesting resources that are super useful for a lot of the analysis that you might be doing, that genomics plus EHR can validate clinical findings and improve early detection, and that we can devise machine learning on the EHR that can identify rare disease or potential rare disease patients. So I'll stop here by acknowledging a lot of the people that have spent a lot of their past 3, 4, 5 years on creating these resources, all the way from the IPH community to the computational medicine community to the CTSI, through folks that are involved in biobanking, from pathology. It really takes a lot of people to create this types of research, all the way to the IT community that has helped a lot on the health IT side. So I'll stop there. I'll acknowledge more folks over here, and obviously the ones to thank the most are the UCLA patients that have consented to be part of all this genetic studies. I'll stop here, and I don't know if there are Q&A questions that I can take. Okay, so the first question is, what's the process of accessing DDR? If you go on the Institute of Precision Health website, there's an entire form there and a procedure that one can follow to get access to both the DDR as well as the Atlas. Okay. For studying neoplasm, is there scope for adding somatic mutation data into the DDR Atlas? And how can we help? Yes, I think there's scope to do that, and I think that'll be a very fascinating thing to do. And maybe we should have conversation about how to do that together with the cancer center and they are and their I think there are some processes in the works about doing that. So that'll be fascinating, and that'll be a tremendous resource to put together. 2 more data or 2 more profile with Atlas and with the DDR, absolutely. So we can. I'm happy to sync with the folks that are working on that end. All right. Regarding phi codes, working with single cell databases, we're working disease ontologies. Do you think phi codes would apply? I think I'm missing. Do you think it will apply to single cell disease ontologies? I'm not completely following this, but the phi codes were created based on human phenotype ontologies. So I think they would be applied to the disease ontology, but maybe I'm missing something there, and I'm happy to talk more about it. Okay, so another question from Manish. So many rare monogenic disorders are not actually Mendelian. Can you comment on complete parentage? What's your take on it? Yes, so obviously I had to simplify things tremendously in the first talk, but yes, you're absolutely right. So as we learn more and more about the Mendelian diseases, we find out that they don't have complete penetrance, and there's an interaction between, let's say, mutations that are coming on this genes, and let's say common variants on other parts of the genome that might influence the penetrance, if you will, or might influence the disease outcome of those patients. So yes, I think that will be super interesting. And in order to analyze or to investigate that, we would need large studies of exome sequencing and common variants. So the common variant part requires lots of individuals to be powerful, and sample sizes are key there. I would also say that there's a lot of work more recently that are observing a lot of overlap, a lot of synergies between the genes that are Mendelian or are found by the Mendelian, they have incomplete or high penetrance, and the genes that are coming up from all these complex traits. So the dichotomy that I started with this talk is not completely warranted. There's a strong overlap in terms of the gene, in the sense that the same genes that get disrupted by coding variants and they lead to Mendelian or severe forms of particular phenotypes can also be just regulated. So they can also be just their transcription output can be just slightly regulated, and they might lead to a more mild form of the phenotype. For example, you can think about genes that cause skeletal dysplasia, and then genes that are impacting height in humans. So there's a strong overlap between them, and putting them together would probably shed some light in this full penetrance versus incomplete penetrance versus very slight penetrance, which comes from the common variant. All right. Any other questions? I think I'll defer to my panel whether we have more time for questions. I guess one more question from Jason. Does the rare disease classifier make use of temporal information of feature onset? It does not. And this is one area that will be super interesting to work more on it. No, at this point, we don't look at temporal information in the feature, but that will be a super interesting thing to look at. I fully agree. All right, if there are no other open questions, I think thank you all for participating, and we'll adjourn, I guess.